Hello everyone, welcome back to Chapter 6. Chapter 6 is all about energy and metabolism. Now basically every task performed by every organism on Earth requires energy. We require energy to perform heavy labor tasks, to exercise, but we also even use a considerable amount of energy during sleep. In the same way that every organism requires energy, every cell requires a constant supply of energy as well. This is because cells and organisms alike live in open systems. An open system means that we are constantly exchanging matter and energy from the surroundings and back to the surroundings. Let's take plants for an example. Plants get their energy from the sun. They use that energy to perform tasks, and then they release that energy in the form of heat. Well, cells do the same thing. The photosynthetic cells in plants receive their energy from the light, the sun. They use it to do cellular activities, and then they release energy in the form of heat. We use the term bioenergetics to describe this concept of energy flowing through living systems. Within a cell, there are thousands of metabolic reactions occurring. Each cell must extract energy and apply it to perform work. The totality of all of these chemical reactions is what we call the metabolism. Every reaction that is happening within your cells and within your body is what is known as your metabolism. All branches of life share some of the same metabolic pathways. Now, what this suggests is that all organisms evolved from the same ancient common ancestor. Now, this ancient prokaryotic ancestor would have came into existence about 3.8 billion years ago. During this time, our atmosphere lacked oxygen. So in order to survive and obtain energy, use that energy and release it, these organisms had to have an anaerobic metabolism, or a metabolism that follows pathways that do not require oxygen. Over time, the metabolic pathways diverged as organisms had to adapt to their new oxygen-rich environment and began to develop new specialized enzymes which better allow them to adapt to their environment. Metabolic pathways begin with a specific starting molecule, and then after a series of steps, each of which is catalyzed by a specific enzyme, you end up with a specific product. Anabolic pathways are metabolic pathways that require an input of energy in order to synthesize complex molecules from simpler ones. Synthesizing large proteins from amino acid building blocks is an example of an anabolic pathway. Other examples include synthesizing sugar from CO2 and synthesizing new DNA strands from nucleic acid building blocks. These anabolic reactions are absolutely critical to the cell. They take place constantly and demand constant energy that ATP and other molecules like NADH or NADPH provide. Now, the opposite of an anabolic pathway is called a catabolic pathway. These types of reactions release energy by taking complex molecules and breaking them down into simpler ones. An example of this would be taking glucose or a fat molecule and breaking it down to release ATP. Now, though these reactions release energy, they also take energy or require energy in order to take place. In other words, they're not spontaneous reactions. Typically, an enzyme facilitates or catalyzes each step of these reactions. Energy is broadly defined as the ability to do work. But energy exists in many different forms. Two of the main types of energy we're going to talk about is kinetic energy, which is the energy of movement or objects in motion, and potential energy, which is the energy that has the potential to do work, like the energy stored in bonds. Once a bond is broken, energy can be released. Now, there are many types of kinetic and potential energy. For example, thermal energy, which is the energy of moving particles or heat, mechanical energy, the energy of objects in motion, electrical energy, the energy of particles moving through a wire, and magnetic energy, which is the energy that causes a push or a pull between magnetic objects, are all types of kinetic energy or movement energy. Now, chemical energy, the energy stored in food or fuel, elastic energy, the energy stored in objects that are being stretched, as well as nuclear energy, the energy stored in the center of particles, and gravitational energy, which is the energy stored in an object when it's falling or when it's above the Earth's surface, are all types of potential energy. Now, the type of potential energy that we're going to focus on is called chemical energy. Chemical energy is the type of potential energy that exists within chemical bonds. The type of potential energy that we're going to focus on is called chemical energy. And again, chemical energy is the type of potential energy that exists within chemical bonds that can be released when those bonds break. A common example of chemical energy that we use every day is the combustion of fuel. The molecules in gasoline contain long chains of hydrocarbons. Once broken, provide energy to your car. All of an organism's metabolic processes and chemical reactions are subject to the laws of thermodynamics. Now, thermodynamics refers to the study of energy and energy transfer involving physical matter. Now, we call all of the matter and its environment relevant to a particular case of energy transfer the system. And everything that is outside the system is called the surroundings. And systems can be one of two ways. They can be open or closed. Open systems are systems in which energy can be transferred between the system and its surroundings. A closed system is a system in which energy must be transferred within the system. Now, as we said earlier, biological organisms are all open systems. Energy exchanges constantly between them and their surroundings. Now, the first law of thermodynamics deals with the transfer of energy in and among all systems in the universe. And it states that the total amount of energy in the entire universe is constant. Or in other words, everything that has always been here will always be here in terms of energy. Now, energy may transfer from place to place or transform into different forms, but cannot be created or destroyed. Now, whether you're looking at an open system or a closed system, no energy transfer will be completely efficient. Every energy transfer loses some amount of energy typically in the form of heat energy. Now, heat energy, defined thermodynamically, is energy that transfers from one system to another that is not doing work. So, for example, when an airplane flies through the air, it loses some of its energy as heat energy due to friction with the surrounding air. The friction actually heats the air by temporarily increasing air molecule speed. Likewise, some energy is lost as heat energy during cellular metabolic reactions. Now, the more energy that we lose from a system, the less ordered and more random the system becomes. Scientists actually measure the amount of randomness or disorder within a system. This measurement is known as entropy. High entropy means high disorder and low energy. Low entropy means there is a high amount of order and a high amount of energy. Now, these energy transformations will proceed spontaneously, meaning that matter will constantly be converted from a more ordered and less stable state to a less ordered and more stable state. So let's think about how entropy relates to the three states of matter: solids, liquids, and gases. A gas would have the highest amount of entropy out of those three because it is the most disordered and most random. A liquid would have less entropy than a gas, but still more entropy than a solid. So now let's think about your room. Over time, if you didn't put any type of effort or energy into maintaining your room, it would gradually get more disordered, because it takes energy in order to put things back in their places and clean the room back up. So an ordered room would have less entropy than a disordered room. Okay, now that we have covered the energy is transferred within a system, we need to talk about how do we measure that energy that is transferred, and how do we compare it with others? Well, the answer is free energy, or Gibbs free energy, which is abbreviated with the letter G after Josiah Willard Gibbs, the scientist who developed the type of measurement. Gibbs free energy refers to the energy that takes place within a chemical reaction or within a system that's available after we account for entropy. Or in other words, Gibbs free energy is the usable amount of energy, or the energy that's available to do work. Every chemical reaction involves a change in free energy, and that change in free energy is called the delta G, which is denoted by this symbol right here. Now, the way we calculate the change in free energy, or the delta G, for any system that undergoes a change is by subtracting the amount of energy lost to entropy from the system's total energy change. Now, this total energy change is what we call enthalpy. Enthalpy is denoted by the delta H, or this symbol here. So when using this formula to calculate Gibbs free energy, you'll take the delta H, or the total change in energy within the system, and from it you'll subtract the product of the temperature in kelvins and the change in entropy. Now, your answer from this equation will be expressed as the amount of energy per mole of the reaction product. So it'll be written in either kilojoules per mole or kilocalories per mole. When using this equation, it is assumed that the chemical reaction took place under standard pH, temperature, and pressure conditions. We generally considered a standard pH to be 7.0 on the pH scale, the temperature to be 75 degrees Celsius, and the pressure to be 100 kilopascals, or 1 atm. As you use this equation more and more, you'll begin to notice a trend in your findings. There will be an inverse relationship present, so that as entropy increases, the free energy will decrease. After you calculate your change in Gibbs free energy, you'll be left with a positive or a negative number. Now, if you have a positive delta G, that means that you have an endergonic reaction, meaning that the products have more free energy than the reactants started with. These types of reactions will have higher enthalpy and lower entropy, meaning that they will have a higher amount of energy in the system and a lower amount of randomness. Now, these types of reactions are not spontaneous, meaning that they require an input of energy in order to take place. This image on the left shows an endergonic reaction. And as you can see, the reactants start pretty low. Quite a bit of energy is added, and you end up with products that have a higher energy state than the reactants did. Now, the difference between where your reactants start and where your products end is the change in Gibbs free energy, or your delta G. Now, the same is true for your exergonic reaction. Even though it looks a little bit different, you're still measuring the change in energy from the reactants to the products. The difference is, in an exergonic reaction, you will have a negative delta G, or change in free energy, because your reactants will have had more energy, or a higher energy level, than your products will end up with. An easy way to think of this type of reaction is that exergonic means that there is energy exiting the system. Now, these types of reactions will also have a higher amount of enthalpy and a lower amount of entropy, meaning a lower amount of energy and a higher amount of disorder. Unlike the endergonic reactions, the exergonic reaction is spontaneous, meaning that it can occur without any energy being added into the system. This image shows a hydroelectric turbine that converts energy of flowing water into mechanical energy. Then, a hydroelectric generator will convert the mechanical energy into electricity that we can use. The turbine on the left represents an exergonic reaction, meaning that the reactants on the left, the water that is moving down to the right, has a higher energy state than the products, or the water that has already moved on into the lower tank. Now, if this system were to continue, obviously it's a closed system because the water's not moving out of it, it'll reach an equilibrium, meaning that the change in free energy will be zero, because none of the water in this turbine can move anymore. In that way, if living cells lived in a closed system, eventually they would reach a point in which no more work could be done. This is because the cell would eventually use up all of the reactants it had, and it would have nowhere for the products to go. That is why cells must exist within an open system, so that there is a constant flow of materials in and out of the cells. This example shows a multi-step open hydroelectric system, meaning that water comes in one end, filters through a series of hydroelectric turbines, and eventually flows out the other end. Through every step, free energy is being released. This is similar to how catabolic pathways within cells work. Each step within a catabolic pathway will release free energy. Of course, a cell is not the only component that exists within an open system. Organisms and ecosystems alike all function within an open system. An organism must capture energy from its surroundings. It'll eat food, which is chemical energy, use it to perform cellular processes, and then excess energy will be excreted or burned off as heat energy. Similarly, an ecosystem must have input of energy. There must be energy coming from the sun, from other surroundings, being transformed, and then eventually being released back outside of the ecosystem. Now, every reaction that takes place needs something called activation energy. The activation energy is the amount of energy necessary for a chemical reaction to occur. It's the nudge that these reactions need to get started. And even exergonic reactions, which are classified as spontaneous reactions, meaning they do not need energy input into the system in order to take place, actually needs a small amount of activation energy. The activation energy is the difference between the peak energy amount and the amount of energy that the reactant started with. And this is the same for both exergonic and endergonic reactions. The activation energy over here is still the difference between the peak energy level and the initial state of the reactants. So within a cell, you might be wondering where this extra energy comes from. Well, the answer is ATP, or adenosine triphosphate. ATP is a relatively small, simple molecule, but within its bonds it contains the potential for a quick burst of energy that can be harnessed to perform cellular work. Think of this molecule as the cell's primary energy currency in much of the same way that money is the currency that people exchange for things. ATP powers the majority of energy requiring cellular reactions. As its name suggests, ATP is made up of 3 phosphate groups bound to an adenosine. Adenosine is a nucleoside that consists of the nitrogenous base adenine and a 5-carbon sugar, ribose. The 3 phosphate groups can be labeled alpha, beta, and gamma depending on their order and distance from the ribose. Alpha being the closest to the ribose and gamma being the furthest from the ribose. The bonds that link these 3 phosphate groups are called phosphoanhydride bonds. These are both equally high energy bonds. Now they are called high energy bonds because the products of such bond breaking between ADP and one inorganic phosphate group have considerably lower free energy than the reactants of ATP and a water molecule. Now, because this reaction takes place using a water molecule, it's a hydrolysis reaction. So in other words, ATP hydrolyzes into ADP in the following reaction. Now this equation shows exactly what we just talked about. When you take an ATP molecule and add water to it, the bond will break in between the gamma and beta phosphate group, which will release 7.3 kilocalories per mole of ATP energy, and you will be left with an ADP molecule and an inorganic phosphate. Like most chemical reactions, ATP hydrolysis is reversible. Being able to quickly synthesize ATP is critical for a cell, because ATP is a highly unstable molecule. Unless it's used quickly to perform work, it'll spontaneously dissociate into ADP and an inorganic phosphate, with the free energy being released as heat. In order to synthesize ATP, you must have an ADP molecule, an inorganic phosphate, and an input of 7.3 kilocalories per mole. Some reactions on their own may be too endergonic to occur naturally. So what the cell does is couple these endergonic reactions to another reaction which is strongly exergonic. Now, a reaction that is commonly used is the hydrolysis of ATP into ADP, or the exergonic reaction that we just spoke about. When the gamma phosphate, or the third phosphate attached to the ATP molecule, breaks the bond connecting it to the other phosphate molecules, that energy is then used to fuel the endergonic reaction in which ATP hydrolysis has been coupled with. Now, one really important example of energy coupling using ATP involves a transmembrane ion pump called the sodium-potassium pump. The sodium-potassium pump drives sodium out of the cell and brings potassium into the cell. A large percentage of the cell's ATP powers this pump because a large amount of sodium is being brought into the cell and potassium out of it. The pump works constantly to stabilize the cellular concentrations of these two molecules. In order for the pump to turn once, one ATP molecule must hydrolyze. When ATP hydrolyzes, its gamma phosphate does not simply float away, but it actually bonds onto the pump's protein. We call this process of a phosphate group binding to a molecule phosphorylation. In a phosphorylated state, the sodium-potassium pump has more free energy and is triggered to undergo a conformational change. This change allows it to release sodium ions to the outside of the cell. It then binds to extracellular potassium ions, which through another conformational change causes the phosphate to detach from the pump. The phosphate release triggers the potassium ions to release to the cell's inside. Essentially, the energy release from the ATP hydrolysis couples with the energy required to power the pump and transport the sodium and potassium ions. So now we're going to talk a little bit about what determines the speed of a reaction and what can actually influence that speed. First, let's take a look at exergonic reactions. Now, an exergonic reaction's rate depends on the activation energy required to initiate the reaction. A larger amount of activation energy will cause the reaction to proceed more slowly, whereas a smaller amount of activation energy will cause the reaction to proceed more quickly. So that is the first way that rate can be influenced or increased in an exergonic reaction, by lowering the activation energy. Now, the way you lower the activation energy is by adding a catalyst or an enzyme, something that speeds up the reaction because it lowered the activation energy. The other way in which an exergonic reaction's rate can be increased is by increasing the energy of the reacting molecules. And the way we increase the energy of molecules is by heating them up and making them more excited. As I mentioned, a catalyst can be used to speed up the rate of reactions. A catalyst is a substance that increases the rate of a chemical reaction without itself undergoing any permanent chemical changes. Both of these images show what a reaction would be like catalyzed and uncatalyzed. It would take about twice as much energy to proceed with this reaction uncatalyzed. Now, the difference between these two reactions is that the left reaction is exergonic and the right reaction is endergonic, both of which have been sped up by the use of a catalyst. Now, the types of biological catalysts that cells use are called enzymes. With the use of enzymes, cells are able to regulate all of their chemical reactions. Virtually every chemical reaction that takes place in an organism is regulated with the use of an enzyme. Now, most enzymes are proteins, though some types of enzymes may be RNA molecules. Enzymes are extremely important to the cell because without them, many of the chemical reactions necessary for cellular life would be too slow to occur in nature without the enzyme. For example, without the use of an enzyme, only 200 molecules of carbonic acid could be made per hour. With the use of an enzyme, 600,000 molecules are able to be formed per second. We can think of enzymes like a little Pac-Man character that's trying to grab up other molecules. Now, these other molecules that it's trying to grab up are called substrate. Now, these are the chemical reactants involved in whatever reaction is taking place. And these reactants will bind to the enzyme at a place called the active site. Now, the active site is the location within the enzyme where the substrate will bind to. The active site performs several different important functions within the enzyme. Number one, it must be capable of orienting the substrate correctly. It must be able to strain the substrate bonds or modify the substrate in whatever way it is meant to. It must provide a favorable microenvironment. And last, it must be able to covalently bond to the substrate for short periods of time. The active site possesses very specific physical and chemical properties that are necessary in order to bind with the enzyme's substrate. The 3D shape of the enzyme must match the substrate exactly. Just like the combination of amino acid residues, their positions, sequences, structures, and properties creates a very specific chemical environment within the active site that if any of this were to be altered, including a change in temperature or pH to outside of the optimal range, then both the chemical and physical structure of the enzyme can be altered or denatured. Once an enzyme has become denatured, it is no longer compatible with the substrate that it is meant to be bound to. So when a substrate actually binds to the active site, it causes a mild shift in the enzyme structure that makes the enzyme fit the substrate perfectly. The time in which the enzyme is perfectly fit to the substrate is known as the enzyme-substrate complex because they are bound. Now, these interactions between the enzyme and the substrate is what we call the induced fit model. Here we see an example of how the enzyme sucrase binds to the substrate sucrose. Once bound in a complex called the enzyme-substrate complex, sucrase will break the bonds between the glucose molecule and fructose molecule that comprise sucrose and release the glucose and fructose. Once the products have been released, the enzyme is free to bind to another substrate. The amount of enzymes present within a cell is not a constant. The cell tightly regulates its metabolic pathways by switching on or off genes that encode for specific enzymes. This in turn regulates the amount of enzymatic activity present within the cell. When a gene is activated, that means more enzyme will be made and the reaction rate will increase accordingly. When the gene is turned off, the enzyme will not be made and the reaction rate will plummet. When a gene is activated, that means that more of the enzyme will be made and the reaction rate will increase accordingly. When the gene is turned off, the enzyme will not be made and the reaction rate will decrease. These two images illustrate what affects the concentration of an enzyme on the left and the substrate on the right bear on the rate of a reaction. On the left, we see that as enzyme concentration increases, a linear increase in the rate of reaction occurs. But on the right, you'll notice that as the substrate concentration increases, the rate of reaction will increase for a while before leveling off. So what this means is that the rate of reaction can be more effectively controlled by modifying the concentration of enzyme rather than the concentration of substrate. For example, if you wanted to increase the rate of a reaction, but you did so by adding substrate to the reaction, for a while this would increase the reaction because there would be more substrate available to the enzymes. But after a while, all of the enzymes would be occupied, so with all of the enzymes working at full capacity, the substrate wouldn't be able to bind any faster. However, the opposite would be true if you increased enzyme concentration. The substrate would remain the same, but there would be more enzymes present in order to bind that substrate, so there would never be a buildup of substrate waiting to be bound to the enzyme. Some enzymes are capable of functioning alone and consist only of one protein component. However, others may consist of a protein component called an apoenzyme, which is basically an inactive enzyme, but they may also require a helper ion or a helper molecule. These can be called coenzymes or cofactors. A coenzyme is an organic molecule that binds loosely to the active site of an enzyme. Once bound, it fulfills the necessary microenvironment needed by the substrate to bind to the enzyme. Now, a cofactor is a metal ion that does not bind to the active site, but rests within it in order to complete the microenvironment needed by the substrate to bind. Inhibitors are another way in which the cell can regulate enzymatic activity. An inhibitor is a substance that binds to an enzyme to decrease its activity. But there are several types of inhibitors. The first would be a competitive inhibitor. Now, a competitive inhibitor is a molecule that is similar enough to the substrate that it can bind to the active site and simply block the substrate from binding. It is called competitive inhibition because the inhibitor molecule competes with the substrate for the active binding site. Now, the opposite of this would be called a non-competitive inhibitor or an allosteric inhibitor. Now, this is a molecule that does bind to the enzyme, but at a site other than the active site. Whenever this type of inhibitor binds to the allosteric site, it causes a shape change in the enzyme that makes the enzyme unable to bind to substrate. You can see in this image that the non-competitive inhibitor is more effective at controlling the rate of a reaction than the competitive inhibitor is, because the non-competitive inhibitor is not having to actively compete with the substrate. The last type of inhibitor I'm going to mention is called the irreversible inhibitor. This type of inhibitor binds with the enzyme and deactivates it permanently. Now, this slide just gives you a better idea of what these competitive and non-competitive inhibitors look like and do. So on the left, we have a competitive inhibitor, which binds to the active site and blocks the substrate. And on the right, we have a non-competitive inhibitor, which binds to the allosteric site, or a site that is not the active site, which then changes the conformation of the active site and blocks the substrate from binding. Now, a lot of examples of these inhibitors include toxins, poisons, pesticides, and antibiotics. An example of an antibiotic would be penicillin. Penicillin is an active site inhibitor for different types of bacteria. So you know that an allosteric inhibitor binds to an allosteric site within an enzyme to deactivate the enzyme. But the opposite of that would be an allosteric stimulant. An allosteric stimulant or activator is something that binds to a deactivated enzyme, modifies the active site in order to open it for substrate, or make it available to bind to substrate. The last method of regulating a metabolic pathway that we're going to talk about is called feedback inhibition. Feedback inhibition involves using a reaction's product to regulate its own further production. The cell will respond to an abundance of a specific product by slowing down the production during its anabolic or catabolic reactions. So this example that we see in the slide shows the end product of a series of reactions becoming an allosteric inhibitor for the original enzyme, thereby blocking the enzyme's ability to bind to other substrates and decreasing the amount of product within the cell. An example of an allosteric regulator is ATP. ATP can regulate some of the enzymes involved in sugar's catabolic breakdown, which is the process that produces ATP. When ATP is abundant, the cell can prevent its further production, which is important because ATP is an unstable molecule that spontaneously dissociates into ADP. If too much ATP were present in the cell, much of it would go to waste. Okay, that concludes Chapter 6. I hope you guys enjoyed it, and I hope you have an awesome day.